Hello, and thank you for joining us for today's presentation, part of our Agilent Cell Analysis Global Conference. My name is Yama Abbasi, and I'm the Vice President of Business Development at ACEA Biosciences, which is now part of Agilent Technologies. And I'll be your host for this presentation. Before I introduce our speaker for today, allow me to share a bit about Agilent, our products, and our products for cell analysis. Agilent, as a leader in life sciences, is very focused on enabling our customers to gain the insights they seek. Our rapidly growing cell analysis portfolio enables deeper, more reliable insights across a variety of cell analysis applications, where investigators and drug developers seek to understand dynamic cellular environments and interactions. This is critically important in the large and growing immuno-oncology and immunotherapy fields. For example, discovering safe, potent, and persistent immune cell products requires more comprehensive understanding of cancer biology and immune cell function. Our presentations today will speak specifically to our solutions and how they are being employed in that context. Our speaker, Dr. Juan Miguel Sánchez Nieto, studied biotechnology at Francisco de Vitoria University in Madrid, Spain. He moved to London in 2010, where he did his master's in molecular medicine at University College London. Juan Miguel continued his scientific career at Imperial College London, where he completed a PhD in developmental and stem cell biology. His research was focused on understanding the differential sensitivity to cell death observed at the onset of embryonic stem cell differentiation. He joined cell and gene therapy catapult industrialization team in 2017 as an analytical developmental scientist. Juan Miguel has been involved in the experimental design and execution of multiple projects related to the development of novel analytics for viral vector, stem cell, and somatic cell therapies. He has been leading cell and gene therapy catapults efforts in the development and qualification of in vitro potency assays for TCR and CAR T-cell-based immunotherapies. I had the pleasure of meeting Juan Miguel last year at Catapult's facilities in London, which was a very impressive facility with some spectacular views of London. Before I hand over to Juan Miguel, a couple of housekeeping notes. There will be question and answer period immediately following the talk. And to help us get as many questions as possible, we encourage you to type in your questions during the presentation. And with that, I will pass it on to Juan Miguel. Thank you.
Hello, everyone. Thank you very much, Yama, for your kind introduction. And also thank you to the teams at Agilent and LabRoots for giving me the chance of presenting at this global event today. Also, I would like to thank you for attending this session, where I will try to share with you some of the results that we obtained trying to develop an impedance-based assay to evaluate the potency of immunotherapy products. But before going into the results, I would like also to take this opportunity to introduce our organization, the Cell and Gene Therapy Catapult. We are part of a world-leading network of technology and innovation centers that are strategically located around the United Kingdom. We were established by Innovate UK as a non-for-profit independent center back in 2012. And it is our vision for the UK to be a global leader in the development, delivery, and commercialization of cell and gene therapies. We can do this by supporting businesses to start, grow, and confidently develop advanced therapies and trying to get them to the patients as rapidly and effectively as possible. We can support these businesses through our development center that is based in central London. Here we have a fantastic professional team that can support analytical characterization activities, process development, and scaling up using different types of bioreactors, we have a dedicated viral vector team and also a stem cell expansion and differentiation platform. In addition, we are developing an integration and collaboration center where we will be able to work directly with our collaborators. We also have a manufacturing center located in Stevenage. Here we have 12 segregated large clean rooms that leading cell and gene therapy companies can use to start manufacturing their products to support their clinical trials or directly for commercialization. In addition, we can support you with different areas such as regulatory affairs, health economics, or clinical support. But if you have any other questions, please don't hesitate to contact us, or you can visit our website for further information. After that brief introduction of our organization, now we can move on with the rest of the presentations on how we have developed potency assays for immunotherapies using the Impedance platform. So with this slide, I would like to highlight what is the current procedure of manufacturing of a T-cell product. The process starts when we receive the patient material into the manufacturing facility, and here it undergoes several steps, such as washing, selection of the material, and then we need to activate the cells to start inducing cell proliferation. A few days after, we use viral vector to be able to insert our transient of interest into the patient material. And then this is followed up by an expansion period of 1 to 10 days that will allow cell proliferation to generate enough material for the patient. The last step consists on washing and concentrating the material and resuspending it into the final formulation. However, before the product is released from the manufacturing facility, it must pass some quality control and release criteria where we need to verify the identity of the product by characterizing the type of cells that we can find on it, what is the appearance, what are the level of impurities. So in this case, that could be the percentage of non-CD3 positive cells. We also have to ensure that the product is safe. So in this case, showing that it's completely sterile or that the levels of mycoplasma or endotoxins are within the acceptance criteria. And also, we need to evaluate the potency of the product. In this case, we can look at different parameters such as the cell-killing activity or cytokine stimulation when the T cells, the engineered cells, recognize their target cells. But one of the important challenges that the industry faces nowadays is to try to reduce this manufacturing process and also the steps between quality control release and the time that it needs to go to the patient. So the current methods that people normally use to evaluate the T-cell potency are based on the Chromium release assay. This assay is the gold standard in the literature, and for those of you who are not very familiar with it, there is a diagram on the right-hand side of the image that tries to explain how it works. The first step would be to incubate our target cells with a radioactive compound, which is Chromium 51. After we have incubated our target cells with this radioactive reagent, we will co-culture them with the effector cells, or the engineered T-cell. When the engineered T-cell recognizes the target cell, it will induce its cell death, and the Chromium 51 will be released to the supernatant. And then we can measure the amount of Chromium 51 released as a way of measuring the potency of the product. However, the use of this type of assay has several limitations. One of them is time. Longer incubation times can lead to Chromium 51 unspecific leakage, and that can make a little bit complicated the interpretation of our results. Another aspect would be the safety one. So we will have to have a dedicated specific area to be able to handle radioactive materials. And another limitation would be cell requirements. This type of assay needs to use normally high effector to target ratios. And so that means that we will need to use many more effector cells to be able to observe a killing effect on the target cells. And if these ratios are very high, then we will lose the physiological relevance of the assay. And another important aspect is that when we are developing these quality control release assays, the idea is to try to use as little as possible of the patient's material, so then the patient can receive as much as possible of the finalized product. So over the years, there has been some developments trying to find alternatives to this Chromium 51 release assay. For example, we could use a kit that is called Cytotox 96. In this case, it measures the release of a cytosolic enzyme, which is lactate dehydrogenase, through absorbance. There are other kits, such as the cell titer glow. In this case, it measures ATP through luminescence. Or we could monitor other release of different dyes that are not radioactive. In this case, an example would be calcein AM or BATDA release as well. And we can also use other technologies such as flow cytometry, where we can monitor cell death or also cytokine release, or other technologies such as Luminex or ELISA, again, to measure cytokine release. However, all these different technologies requires pre-labeling either the effector or the target cells to be able to identify the killing ratios. Or another disadvantage is that we are not able to obtain any information about the kinetics or the dynamics of the killings. So is the operator the person that needs to determine which is the optimal time point to be able to observe these killing effects. So one potential alternative to be able to overcome some of these limitations is to use an impedance-based potency assay. This platform is a real-time cell analysis system that is non-invasive for the cells and is based on electrical impedance. So it's just a small electrical pulse that will go through the cells, but it doesn't affect their identity or their viability. It's a label-free assay, so we don't need to label either the effector or the target cells, saving time and reagents. The platform that we have at our organization offers high throughput because we can put up to 6 96-well plates that give us the possibility of testing many different conditions at the same time. And it's also very flexible because each of the 96-well plates is independent from each other. So different operators can start different experiments at their time of their convenience. However, it has also some limitations, and we need to optimize the process for each specific target cell line that we are using. So how did we decide to use this platform in our organization to evaluate the potency of engineered T cells? We followed two different strategies. The first one was to try to measure the potency for a T cell receptor-based therapy or a TCR-based therapy. And in this case, we evaluated the use of either non-adherent target cells or adherent target cells. And the second strategy was to try to evaluate the potency for a chimeric antigen receptor or CAR-T cell-based therapy. And in this case, we only used adherent target cells. But how does the impedance potency assay work? So for those of you who are not very familiar with the instrument, you have a picture of it on the left-hand side of the slide. So we have the laptop with the software, and then on the right-hand side, you can see the part of the unit that has the platforms for the 6 96-well plates. And on the bottom, you can see how the special plates that we need to use for this instrument, that are called E-plates, look like. Hopefully, you can appreciate that they are coated with gold microelectrodes that then are going to be used to measure the electrical impedance. So for the assay to be able to work, we need to have a conductive solution, such as the cell culture medium. And then once we apply a small electrical current, the electrons will be able to flow from one electrode to the other. And just in the presence of medium, the electrons can flow freely. So there is nothing that is going to block their flow. And that is interpreted as a zero cell index. Cell index are the arbitrary units that we use to monitor impedance. Once we add our target cells, the cells will start to attach. And once they attach, they will block or impede this electron flow. and that is translated as an increase of the cell index. The more cells attached, the higher the cell index will be, and then once we have a maximum attachment, the signal will reach a plateau. And that is a specific time point that we are looking to be able to add our effector cells. Once we add our effector cells, they will recognize the target cells, they will kill them, and these target cells will die, detached from the plate, and that killing effect is translated as a decrease on the cell index. And that's how we can monitor in real time the potency of our T cells. So let's move on and see what are the steps that we follow trying to develop this assay for a TCR-based product. So before just starting to explain our results, let me just try to introduce a little bit how the TCR-mediated killing occurs. For the T cell to be able to recognize and kill the target cell, we will need two different signals. The first signal is given by the interaction of the T cell receptor, or TCR, and the major histocompatibility complex 1, MHC1, loaded with the antigen of interest. So this recognition is the first primary signal. However, this is not enough for the T cell to kill the cancer cell. We need a secondary signal that is given by different co-receptors. In this case, this is represented by CD28 and 4-1BB, but there are many others. So depending on the expression of the co-receptors and the interactions that they get, the response of the T cell would be modulated. So once the T cell receives these two signals, so the TCR with the MHC loaded with the antigen of interest and the co-receptors, the downstream signaling cascades will induce changes in cell metabolism, cell proliferation, and cytokine secretion that will ultimately lead to the targeting and killing of the cancer cell. So what are the steps that we follow to develop our assay? The first step was to select a suitable target cell line. And in this case, we chose 3 different adherent cells. Cell line 1 and cell line 3 were melanoma cancer cells, and in the literature it was published that they expressed our antigen of interest. Cell line number 2, it was an ovarian cancer cell line, and in the literature it was reported that it does not express our antigen of interest. So we could use cell line 1 and cell line 3 as positive controls for the assay, and cell line number 2 as a negative control. In addition, for some of our quality control experiments, we use a control cell line that was used previously in the literature. Then the next step was to evaluate the optimal seeding density to perform the killing assay. And we also needed to verify that the cells express the MHC class 1, and for that we use an HLA-A2 antibody. And after these steps were optimized, we proceeded to perform the assay qualification. The things that we evaluated were the instrument's linearity. So in this case, we wanted to find out if there was a direct correlation between the number of cells that we seeded and the cell index. Then we also wanted to see if there was also a correlation between the cytolytic potential of our cells and the cell index. And for that, we used 2 different parameters. The first one was the killing time 50, which is the amount of time that we need to be able to reduce the normalized cell index from 1 to 0.5, so basically to reduce it by half. And then the second parameter was the cell index at a specific time point. This was determined by the operators. And the last parameter that was evaluated was precision, so the scatter or the spread of the results with different measurements performed by one single operator and how it compared to the measurements taken by a second operator. So here are the results of the evaluation of the optimal seeding density for each of the cell lines. In this case, we used 3 different densities: 12,500, that is in color blue, 25,000 in red, and 50,000 cells per well in green. And as you can see at the beginning of the assay, there seems to be a correlation between cell index and number of cells. So the more number of cells that we put, the higher the cell index would be. And we managed to get cell index around 1, which is the value that we were looking for, as recommended by the manufacturer, to be able to perform this type of assays with the lower cell density that we tested. And towards the end of the assay, we were not able to distinguish any differences between 25,000 cells or 50,000 cells. So it's likely that at this point, the cells were overconfluent on the well. And in addition to this, we also tested if we could improve cell attachment and adhesion by recoating the wells with fibronectin. And as you can see, we managed to improve the speed of attachment and the strength of attachment because the cell index increased dramatically when we recoated the wells with fibronectin. You can clearly see that if you look at the blue line, which is the lower cell density that we tested, just pretty much a little bit after seeding, the cell index almost reaches a value of 2, whereas in the wells that was not with fibronectin, we were not even reaching cell index 1. The second cell line number 2, gave us a completely different profile of cell index. So that's why it's critical to optimize the cell seeding densities for each of the target cell lines that you're going to use during your experiments. As you can see, these cells attach in a stronger way because the cell index reaches values of around 7 with the lower cell density, which again is in the blue color. 25,000 cells attach really, really fast, and we managed to observe this plateau around 250 minutes after seeding the cells. And something that was quite interesting was to see that in the higher cell density, 50,000 cells per well, we saw a very strong attachment at the beginning, but as you can see, over time, the cell index decreases. And we believe what happens is that the cells were overconfluent at the time of seeding, and then there was not enough nutrients in the medium, and the cells started to die, and that was translated in a decrease of cell index. The use of fibronectin, again, improved the cell attachment. As you can see with the lower cell density in blue, the cell index reaches higher values compared to the ones in the absence of fibronectin, and the signal is more stable. But we still observe the same effect with the higher cell density, 50,000 cells. We can see the decrease of cell index over time due to the overconfluency. And surprisingly, with our third cell line, we didn't reach cell index 1, which is the value that we were looking for, with any of the cell densities that we tested in the absence of fibronectin. However, when we coated the wells with this recombinant protein, we managed to improve the attachment and adhesion of these cells dramatically. And with the lower cell concentration, we managed to get to cell index 1, as we were expecting. The next thing that we did was to evaluate the expression of the MHC class 1 complex, which, as you remember from the previous slides, is required for the TCR-mediated killing. And in this publication, what they did was to pulse or to incubate with increasing concentrations of peptide their target cells, and they saw that this increased the expression levels of the MHC class 1, and they measured this using an HLA-A2 antibody. So we wanted to follow the same strategy to see if by pulsing the cells with our antigen or peptide of interest, we could increase the expression levels of the MHC class 1, and therefore trying to increase the chances of being able to observe a more clear killing effect using the impedance platform. So then what we did was to try to replicate what they did in that publication. So for that, we incubated our cells with our peptide of interest, and we managed to replicate the results. So using their control cell line, we observed an increase in the expression levels of HLA-A2 when the cells were pulsed with the antigen of interest. However, this effect was not observed in any of our adherent cell lines. Cell line number 3, though, showed very high expressions of HLA-A2. Cell line number 1, little expression of HLA-A2, and we were not able to detect the expression of this protein in cell line number 2. But it was not very relevant at this point because that cell line is the one that we were using or planning to use as a negative control, given that it was reported in the literature that it didn't express our antigen of interest. The next type of experiment that we did was to see if there was a negative effect on the cell index when we co-culture the cell line number 1 and cell line number 3 in the presence of non-transduced cells. So in here, what we wanted to evaluate was if the non-transduced cells had a negative impact on the readout of the impedance assay in the presence of the target cells. And you can see that there was a slight reduction in the cell index for both of the cell lines, but we didn't see a dramatic decrease, and the normalized cell index never went underneath 1, which is if it goes underneath 1, then it would be a sign of cell killing. So we decided to move forward with cell line number 3 to keep developing the potency assay. As with the lower cell density, we managed to get a cell index of around 1. It was the cell line that showed highest expression levels of HLA-A2 among the cell lines that we tested. And it had a relatively more constant cell index over time compared to the other cell lines, showing a little bit less of non-specific killing. So the first thing that we did was to try to evaluate the instruments in linearity. So this is trying to see if there is a correlation between the number of cells that we plated and the cell index values. So for that, what we did was to see different cell concentrations with an increased constant cell number. And then we evaluated the cell index, all the values that are in between the black dotted lines. So here you have the results of the first linearity experiment, and you can see that the R-square value, the coefficient of correlation, is very good. It's over 0.97, indicating that there is actually a direct correlation between the number of cells that we have plated and the cell index. However, on the second experiment, even though the R-square value is still very good, again over 0.97, you can see that the cell index values were slightly higher compared to the first experiment. And in the third replicate, we obtained values that were closer to experiment number 1. This slide just wants to highlight the sensitivity of this platform of the impedance-based assay to detect different cell densities once we plate them on the E-plates, and how important it is to be consistent throughout all the process, from detaching the cells until they are seated on the impedance plates. So this requires different manipulation steps, such as detaching the cells from the vessel they were in, performing cell counts, spinning the cells down, and then resuspending in the desired concentration. As an example, if the cell dissociation was not great, and then we have small cell clumps, then these clumps will affect the overall cell counting results, and we will not be seeing the amount of cells that we thought we were going to have. So once we have shown that we can establish a linear correlation between number of cells plated and cell index, then we decided to move on and actually planned experiments to be able to evaluate the cytolysis and see if there is a direct correlation between cytolysis and cell index. So for that, the approach that we took was to fix an effective to target ratio. In this case was a 2-to-1 effective to target ratio. If you remember, the optimal seeding density that we decided to use was 12,500 cells, and therefore we will need a total of 25,000 T cells for this type of experiments. And then we decided to mix transduced, or basically killer cells, with non-transduced cells in different proportions to see if we can find a correlation between the cytolytic potential and the cell index. And in order to be able to measure this correlation, we evaluated 2 different parameters. The first one was the killing time 50, which is represented by the red dotted line. And the killing time 50 is the amount of time that we need to reduce the normalized cell index from 1 to 0.5. And the second parameter that we evaluated was the cell index or the normalized cell index at a specific time point. So for this type of assay, what we do is to normalize the cell index to the point where we add our T cells. So in this case, it's on the y-axis, the time 0. So when we add the T cells, that is the normalization point. And as you can see on the graphs, when we don't have any transduced material, which is the top like light orange color, there is no killing effect as the normalized cell index keeps over 1. When we add some transduced cells, when we have 5% transduced cells, which is the black line, there is some reduction on the cell index compared to the previous condition, but still we don't see any active killing as this normalized cell index keeps over 1. Only when we start to have more transduced material at 20%, 40% is when we start observing a more dramatic killing effect. And it is more apparent with the large amount of transduced material, transduced cells. So as you can see on the blue color, when we have only transduced cells, the cell killing occurs very, very fast, and we manage to almost go to normalized cell index 0. So that would mean almost killing all the target cells that were plated. So in these graphs, you have the results of the two different parameters, the killing time 50 and the cell index, in this case, time 420 minutes. And we found an exponential correlation for experiments 2 and 3, not for experiment 1 in this case. As you can see in experiment number 1, they pretty much have the same killing time 50 values independently of the amount of transduced material. But for experiments number 2 and number 3, we observed a reduction in the killing time 50 with increasing concentrations of transduced cells. And when we look at the graph on the right-hand side, so the cell index at a specific time point, we can observe that the more transduced material that we have, the lower is the value of the normalized cell index, indicating higher killing ratios. And again, we also managed to observe an exponential correlation for this analysis. The next thing that we did was to try to prove the specificity of the product. And for this, we took two different approaches. We compared first what are the differences between co-culturing our target cells in the presence of transduced or non-transduced cells. And this was performed by two different operators. And you can see that we can only observe a dramatic active killing when we use transduced cells. That is the red line on the graphs. And this effect is not observed in the presence of control T cells. And the other experiment that we did to prove the specificity was to use the cell line number 2 that we had. So if you remember, this cell line is the one that does not express our antigen of interest. So in theory, they are not susceptible to be recognized by our T cells. However, in here, we cannot distinguish if we didn't detect the killing due to the absence of our antigen of interest, or because the cells don't express the MHC class 1. Because if you remember, we were not able to detect this with the antibody that we used. But as you can see in the graphs, when we use the transduced T cells, we were not able to observe any killing. And the last parameter that we tested was the intermediate precision and repeatability. So in this case, two different operators performed experiments with transduced cells. And what we did was to see how was the spread of the different repeats that they performed, evaluating the killing time 50. So as you can see, the three replicates for operator number 1 are very close to each other. And the repeats that operator number 2 perform are also very, very good. And there were no significant differences in the values that each operator obtained when evaluating the killing time 50. When we plotted all the different experiments together, we observed that we managed to reduce the normalized cell index from 1 to 0.5 in about 2 hours only, with a standard deviation of plus minus 20 minutes. So this was really, really good because it was a really fast, a reliable, and robust method to be able to measure the cell killing of our cells in a rapid way and very consistent, which was great to see. Then the next step that we took was to see if we could use this platform when we had non-adherent target cells. And this could be a little bit counterintuitive because in order for this assay to be able to work, we need to attach the cells to the bottom of the plate. So in order to try to achieve this, we use two different antibodies, antibody A and antibody B. Antibody A is on the left-hand side and antibody B is on the right-hand side. And to test this, we plated different seeding densities and we used different amount of antibodies. And as you can see from the two plots, antibody B was much better because we managed to obtain a higher cell index when we pre-coated the wells with this antibody. Excuse me. So once we have determined the specific antibody concentration, the next step was to try to identify what was the optimal seeding density. So for that, again, we coated the wells with a specific amount of antibody, and then we tested different seeding densities. And as you can see, all the seeding densities that were higher to the one that is highlighted on the square had the same cell index levels. So therefore, that indicated that we were reaching saturation levels with that specific cell concentration. And it's the one that we decided to use for further experiments. So we have determined the optimal antibody concentration, and we have also determined the optimal seeding density. So once those two parameters were set, we were ready to perform the killing assay. And in this case, the assay outline was a little bit different. What we had to do was to incubate these non-adherent target cells with a specific antigen of interest for 2 hours before plating. Then the target cells are plated and allowed to attach for 4 hours. And as you can see, there is an initial attachment, and then during those 2 to 4 hours, the cells reach the plateau. And this is the point that we were looking to be able to add our effector cells. And then we added different effector to target cell ratios. So 1:1 in blue, 2:1 in purple, and 5:1 in green. So as you can see, the higher the effector to target ratios, the more pronounced is the killing effect. And we were quite interested actually in seeing how this impedance-based platform performed compared to other technologies such as flow cytometry or quantitative image analysis. So in order to be able to compare the different technologies, we designed the following experiment. So we have our engineered effector cells, and then we have our target cells in 4 different conditions. The first condition is the cells that were incubated in the presence of our specific antigen of interest, and then we have 3 negative controls. So that is the target cells alone, and then the target cells that were pulsed with non-specific, 2 different non-specific peptides, and that is represented in the green and in the blue color. After the cells were pulsed, we co-cultured them on the impedance plate in a 5:1 effector to target ratio. And as you can see in the graph, we only observed a killing effect on the cells that were only pulsed with the specific peptide. And from this graph, we could determine that after 8 hours, more or less, there was a clear difference between the positive and the negative controls. And we decided to take that specific time point for our flow cytometry comparative analysis. So in order to be able to perform flow cytometry and distinguish the 2 different populations, we had to pre-label one of them. So in this case, we pre-labeled the effector cells with a fluorescent compound. After the cells were labeled, then we put in co-culture the target and the effector cells. And after 8 hours, we evaluated the levels of apoptotic cells using annexin 5 and 7 AD, and then monitor the proportion of live cells. And as you can see in this chart on the right-hand side, we have in blue the data from the impedance platform, and in orange the data that we obtained from flow cytometry, and they gave very comparable results, where we only see a clear reduction in the viability of the cells when we look at the cells that were pulsed only with the specific peptide, not in any of the other conditions. And we did a similar experiment using quantitative analysis. For that, we have to label the cells, the target cells, with a green fluorescent compound. The effector cells were labeled in blue, and the dead cells will appear later on during the killing in red. And as you can see on the right-hand side, the results of this quantitative image analysis showed very comparable results to the graphs that we have shown in the previous slide with the impedance data. So to summarize this part, we have shown that we can show and measure the potency of a TCR immunotherapy product using the impedance-based platform. We have also shown two different methods to monitor the T-cell cytotoxicity, either using adherent cells and non-adherent cells, and discuss the different parameters that needs to be optimized during this process. And also we have shown that the assay readout correlates with flow cytometry and image analysis. However, the impedance offers some benefits that is label-free and also provides kinetic information of the cell killing. For this last part of the presentation, I wanted to use this example where we supported one of our collaborators trying to progress their product from early research stages into the clinic. And for that, we used a Chimeric Antigen Receptor T-cell product. And the aim of this part of the work was to try to evaluate the potency using the impedance platform. Chimeric antigen receptor or CAR T cells work in a slightly different way compared to the TCR ones. So in this case, the chimeric antigen receptor has 4 different functional domains. We have the target binding domain that is extracellular. Then we have a hinge domain that is a transmembrane domain. And we have 2 intracellular domains, the co-stimulatory one and the essential for the activation. So once the target binding domain binds to the target antigen, that will trigger the cytolytic activity of the T cell, a cytokine release, and cell proliferation. And I am sure that most of you are aware that there are currently 2 FDA-approved medicinal products based on CAR T cells to tackle B-cell malignancies. So, however, if we want to target solid tumors, we have different challenges that we need to face. One of them is access to the tumor site. Another challenge is the lack of appropriate target antigens. We need to find markers that are only expressed on the cancer cells and not in any healthy tissues, because if those were also expressed in healthy tissues, the CAR T cells will also damage them. And another aspect is to consider the immunosuppressive tumor microenvironment. So we thought that a potential therapeutic approach could be to try to target the tumor vasculature. So during tumor formation and tumor development, the tumorigenic cells are able to secrete angiogenic factors that will promote the formation of new vessels. Those new vessels will supply nutrients from the blood, and they could also be a way of metastatic spread. So if we were able to target this new vasculature, we will stop the supply from nutrients to the tumor, and that will definitely slow the growth and potentially be able to reduce the size of the tumor, or maybe even to eliminate it. And we will be able to trying to prevent the metastatic spread of those tumorigenic cells. So our collaborators identified that CLEC14A could be a potential tumor endothelial marker, because, as you can see on this slide, it is only expressed in the vasculature of tumor tissues that we have on the left-hand side of the panel, but not in their healthy counterparts on the right-hand side of the panel. So basically we have on the top breast carcinoma samples, in the middle liver carcinoma, and on the bottom panel bladder carcinoma, and you can see colocalization of CLEC14A and endothelial marker. And however, we are not able to detect CLEC14A on their healthy counterparts in any of the tissues that they evaluated. Then our collaborators in Turin performed some in vivo studies. In this case, they used a RIP-TAC2 mouse model. So these animals developed the tumor in the pancreas, and they used either mock transduced cells or cells that were transduced with the chimeric antigen receptor. And as you can see, in those animals that were infused with the CAR T cells, there was a reduction in the expression levels of CLEC14 in the vasculature. And they quantified these images, and they saw a significant reduction in the vessel area in those animals that received the CAR T cells. There was also a significant reduction in the expression levels of CLEC14 in the vasculature of those animals that received the CAR T cells, and there was an increase in the apoptotic marker caspase-3, which is a marker of cell death, in the vasculature of those animals that received the CAR T cells. In addition to this, they also measured the tumor size, and they found a significant reduction when comparing the tumor volume of the animals that were injected with the mock cells compared to the ones that received the CAR T cells. So there was a reduction of around 50% in tumor volume, which was really promising. However, our challenge here was to try to change a little bit the whole process. So we wanted to try to develop a closed system where we could try to reduce the time of manufacturing and also the way of generating these CAR T cells. So if you remember from the first slides, we discussed that this is a lengthy process that normally takes around 10 to 12 days, and it requires the use of viral vectors. Excuse me. So we wanted to take a different approach and see if we could reduce the overall manufacturing time, avoid the use of viral vectors, and then by these two different strategies, try to reduce the costs of producing this material, and also take a different approach by not having a stable expression of the CAR, but just like a transient expression. And we did that by electroporating an mRNA instead. So we used this instrument, the ClinMax Prodigy, where we can start with the initial material, then we can perform the CD4 and CD8 isolation. We can keep the cells in culture if we need. We can, in addition to perform our electroporation to insert the mRNA into the cells, and then finalize the product with harvesting the material. We tested two different methods, so activating the cells as the typical manufacturing process that we discussed at the beginning of the presentation, and using non-activated cells, and this was the approach that could potentially shorten the whole process. And as you can see, we managed to get very good viability in both cases, and very good transfection efficiency. So it was slightly higher in the activated cells, but it was not significantly different compared to the non-activated material. But for the purposes of optimizing the impedance assay, we used transduced cells. So these were generated with a viral vector, and they were stably expressing the chimeric antigen receptor. So as you can see on this graph, we used very low effective to target ratios. So in this case was 0.5 to 1 and 1 to 1. And even though they were very small effective to target ratios, we managed to observe a killing effect within 3 hours. So on the right-hand side, you can see the quantification of the results evaluating the killing time 50 on 3 different assays. And they were very, very consistent and very reproducible. And as you can see, within 4 hours, we managed to reach the killing time 50 value. But what happened to those cells that we generated with the messenger RNA? So in this graph, we have a summary of the experiment that we performed, in which we have transduced cells, which are our control. So these cells should be inducing a high killing effect constantly over time. And that is what happens. So as you can see on the purple line, over a 9 days time period, the normalized cell index was constantly very low, which is a sign of a high potency of the product. And if you look at the green line, which is the transfected cells with the messenger RNA, so on day 0, they have the same killing potential as the transduced cells. However, over time, they start to lose this potency or this ability to kill because, of course, the messenger RNA gets degraded, and therefore the expression of the Chimeric Antigen Receptor protein will be degraded with the time. And another way that we measure the potency of those transfected cells was by measuring cytokine release, in this case through the Luminex platform, where we could measure the expression levels of interferon gamma and granzyme B. And as you can see, there was a dramatic increased expression of these 2 cytokines when the cells were transfected with the messenger RNA. So this was another additional way to prove the potency of the cells that we manufacture using this new strategy. So to summarize the whole presentation, we have shown that we can measure the potency of either TCR-based therapies or CAR T-cell-based therapies in a reliably and robust way using the impedance spectroscopy. We have shown the specificity of the assay independently of the therapy used. And in addition, again, the impedance assay is label-free and provides dynamic or kinetic data of the cell killing, not only during the assay, but also, as we have seen in the previous slide, over a different period of time. And the assay provides a fast and high throughput alternative to current methodologies that can eventually help us to try to reduce the time between a quality control release and administration into the patient. I would like to, of course, take this opportunity to thank all the people that were involved in these different aspects of this work, either performing the in vivo experiments or in the process development, trying to generate either the transduced or the electroporated cells, members of the team for critical discussion and ideas. And of course, I would like to take this opportunity to thank you. And I hope that you found this webinar interesting and that you can take some ideas to apply to your own research. If you have any questions, please, I would like to have the opportunity to answer them, and I will try my best to do so. Thank you so much again.
Thank you very much, Juan Miguel, for that excellent presentation. We will now start the question and answer portion of the webinar. If you have any questions you'd like to ask, please submit it. We'll be able to get to a couple of questions before the end of the hour. However, even if we don't make it to your questions, we'll be forwarding all of them onto our speaker and the Agilent team to follow up with you. So let's get started with some of the questions to Juan Miguel. So Juan, you talked about basically coating with fibronectin. Is that required all the time in this assay, or maybe perhaps for only certain cell types?
It's only for certain cell types. So some of them we didn't have to use fibronectin, But as you saw in some of the slides, fibronectin really helped to improve the attachments and the cell index signal for some of our cell lines, but it's not required at all times. So it's an optional step that you can perform on your experiments.
Great, thank you for that. And this real-time cell analysis assay, obviously you mentioned that you would have to add the effectors and allow the experiment to proceed. Is there an optimal time point or time duration, let's say, for which this potency assay needs to be conducted, or does that really depend on the type of effector one is looking at?
Basically you answered the question. Is more dependent on your effector cells. Obviously the duration time is going to be affected by your transaction efficiency and also the total amount of cytotoxic T cells that you have in your product. With some of these linearity studies that we perform, we try to show that you will have a faster killing ratio if you have an increased number of cytotoxic T cells.
Okay, great. Thank you. You spoke about two types of parameters when you were doing your calculation. One was the KT50 value, and the other one was cell index at a given time. Do you have any preference for one or the other, or do you think both of those are important, or does each one have a certain unique value?
My personal preference would be the killing time 50 because it's a more consistent parameter. So it will be specific just to measure when you reduce this normalized cell index from 1 to 0.5, that will give you a specific value all the time. Whereas if you choose to determine a cell index at a specific time point, it's more variable and it's just depending on the operator. But it's great because the assay is so flexible, and then depending on your needs, you can choose which parameters suit your needs better. Sorry.
Great. Thank you for that. In your discussion, one of the questions that we got is you talked about like the residuals as part of the linear regression analysis. Can you please expand on that?
So it was one of the parameters that we evaluated to basically see if the linear regression model fitted our data points. So when we evaluated the linearity of the number of cells that we plated onto the impedance and see we have a direct correlation between the number of cells and the cell index, they followed a linear regression because the residual plots show like scatter points. However, when we evaluate the linearity on the cytotoxic potential of the T cells, it was not on the slides, but we also did the residual plots. And in that case, when we did linear regression, the residual plots show the specific pattern. And that means that it's not a model that is going to fit your data points. And when adjusting it to a exponential regression, then the residual plot was scattered, suggesting that our model fitted better an exponential regression for that correlation.
Okay. Well, with that, we are very close to reaching the top of the hour, even though we have some addition for you, but we'll make sure that we will send that to you, Juan. And thank you again for sharing your work with us today. And thanks to our audience for your questions and also for your participation. Thank you very much, Juan.
Thank you very much. Bye.